13 Universal. This is A-level Caie biology, and we're going to be looking at nucleic acid and protein synthesis. Series 1. This is schedule. Come on, let's do this. The first question there says, you can see that co-transport protein and membrane proteins found in companion cells of phloem tissue. Okay. Says messenger RNA is the molecule in cells that carries genetic information, the DNA that codes for co-transporter protein to the sites of protein synthesis in the cytoplasm. Complete table 6.1 to compare the structure of a molecule of mRNA with the structure of a molecule of DNA. So this is table 6.1. You can see that. So we have features, names of bases. Okay. I want you to pause the video and try to solve this question, right? So what are the names of bases you find in mRNA? Okay, what are the names of bases you find in DNA? What are the names of pentose sugars here? And pause the video, do it by yourself, and don't check the mass scheme yet. Okay, so there are 4 bases in mRNA. They include guanine, cytosine, adenine. It doesn't have thymine and uracil. So guanine, adenine, cytosine, and uracil. So this has guanine, cytosine, guanine, adenine, cytosine. Instead of uracil, it has thymine. So this has thymine, while it's uracil, but they have adenine, cytosine, and guanine, right? So name of pentose sugar here is ribose. This is deoxyribose. Number of strand, this is just one strand, this is double strand. Okay, if you check this, you'll see that. Okay, so you see, so they all have adenine, cytosine, guanine, adenine, cytosine. The difference is that mRNA has uracil and this has thymine. Okay, so the pentose sugar here is ribose. This deoxyribose is one single, this is two. There's one strand, 2 strands. Okay, let's quickly go to the next question. Just 3 marks. See, Fig 6.1 shows the events that occurs in the nucleus of a companion cell in phloem tissue to synthesize molecules of mRNA. That's a typo. You can see what's happening here. Okay, this is DNA. Okay, undergoing this reaction to give us another strand of molecule. This molecule undergoes to give us this messenger RNA. Okay, so there's this functional messenger RNA. This is Fig 6.1. So what is happening from here to here, and what is happening from here to here? So let's find out. Name stage 1 shown in Fig 6.1. So I wanted to name stage 1 quickly. All right, name stage 1. Yes, so stage 1, pause the video and name it. Okay, if I'm using DNA to form a single-stranded molecule, this should be transcription. Is that okay? So DNA is used to synthesize RNA, but the first product is primary transcript before something happens to form mRNA. Okay, so that's transcription. Quickly, let's run. Okay, Fig 6.1 shows the event that occurs. Okay, I think the same thing. So the next question says, describe what happens at stage 2. Shown, this is stage 2. What's happening here? So what happens? This is primary transcript. Okay, shown in Fig 6.1 to shorten the length of the RNA molecule. So what happens to primary transcript to shorten? Of course, that is what we call splicing. That's a post-transcriptional event where introns are non-coding, they are removed. Okay, introns are cut off and the exons are joined, and then we have the functional mRNA. Okay, exons are coding, introns are non-coding. Look at it. So removal of introns from primary transcript. So this is primary that's the first one formed, primary transcript. So when we remove introns and we join exons, we form functional DNA and RNA, sorry, and that is called splicing. Okay, all right, so that's 2 marks. It said co-transporters molecules are proteins produced in companion cells. So they are made in companion cells. Okay, it's a fig. That fig there shows what happens in the cytoplasm of companion cells to transfer RNA molecule before the co-transporter protein can be produced. Let's see what happens. So you can see that. So this is transfer RNA molecule. The CCCG, this is anticodon. All right, it has two sides, the anticodon and of course CCA site. This is where amino acid is attached. Can you see the amino acid is attached to this? Do you remember the name of the protein that attaches the corresponding amino acid, all right, to the specific tRNA? Do you remember? Amino acid tRNA, okay, synthetase, okay? Yep. So this is what requires ATP in protein. Okay, synthesis. Translation. Okay, this is post pre-translational event. Okay, let's go to the question. Describe the role of the transfer RNA shown in Fig 6.1 in the synthesis of a co-transporter. So what is the role of transfer RNA? Pause the video. Okay, try to see if you can attempt it by yourself. All right, describe a sequence of events that happens. When they say describe, tell us what and what happens. You can see that here the amino acid is attached, okay, to the transfer RNA, okay, and that attachment is based on the anticodon, right? So the amino acid must be coded by the anticodon. So once it's attached, then the transfer RNA, okay, transports the amino acid to ribosome, where ribosome, of course, it binds to the tRNA. When it gets to ribosome, it then binds to the mRNA on the ribosome using the anticodon by complementary base pairing. Okay, so peptide bond is formed between, I think I need to show you that. Okay, so we're going to get to a point where we're going to require that. So if this is transfer RNA, all right, this is transfer RNA carrying an amino acid here, okay, and this is another transfer RNA, all right, carrying another amino acid. Both of them are binding, they are attached to mRNA. So mRNA is attached to ribosome. Don't forget that, you know, whatever that is here, this is complementary base pairing. So if we have A here, okay, we have C here, we have G here, right? It will bind to its codon and complementary codon, which is going to be U, okay, and then this is going to be G, all right, and that's going to be C. So there's the codon on messenger RNA binds to the anticodon and transfer RNA. You can see, so you can see what is happening here. The same thing happens here, but the most important thing is that when transfer RNA arrives at the ribosome, okay, and then they both bind to the mRNA, then peptide bond is used to join the amino acids, okay, on this adjacent and transfer RNA. So that means that transfer RNA transports amino acid to ribosomes so that primary structures of protein can be formed. You can see that here. So to start from the beginning, reference to amino acid activation or charging amino acid before attachment. So amino acid is charged first, okay, it's called amino acylation. So tRNA attached to specific amino acid. So every tRNA binds to a specific amino acid because their anticodon determines the amino acid they bind to. This is very important. All right, so idea that anticodon, which is CCG here, identifies specific amino acid, helps you to identify it. Then tRNA transports amino. We are describing the role. It transports the amino acid to ribosome. tRNA entering the site of ribosome enters, and then this is anticodon. Anticodon binds to mRNA codon, like what we said here. The anticodon of tRNA binds to codon on mRNA by complementary base pairing. You know, all right. Okay, C, A binds to U, C binds to G, and then of course specific amino acid correct position. So it makes sure that amino acids are in correct position, and it helps to form the primary structure, which is a sequence of amino acid that forms the polypeptide. Don't forget what I said. Amino acid transfer RNA synthetase is the enzyme that joins amino acid to transfer RNA. Okay, and when it happens, we now form amino acid tRNA complex. So this is called amino acid tRNA complex. Okay, the enzyme that joins them is called amino acid tRNA synthetase. Okay, so guys, let's go to the next question. So outline the role of co-transporter proteins in companion cells. So I want you to pause the video and try it. What does co-transporter do? All right, remember that co-transporter, pause the video first and try it. The word co-transporter, co means that it transports two, all right, different molecules. All right, so it transports sucrose and hydrogen ions from the cell wall of companion cell into the cytoplasm of companion cell. So sucrose moves from lower concentration to higher concentration, okay, and that is called secondary active transport anyway. Why hydrogen ion is moving by facilitated diffusion? Okay, so it helps to transport sucrose. Look at it here. Let's know movement of sucrose with proton. Yeah. All right. Sucrose moves from cell wall, apoplast of mesophyll cell. Sucrose moves against concentration gradient. All right, because it says outline, so at least the major effect needs co-transporter protein because sucrose is polar. Yes, that's one of the reasons why it needs it. It's polar, so cannot pass through the membrane. Maintain concentration gradient from sucrose between companion cell and sieve tube. All right, that's one of the things that happens. So sucrose moves into the companion cell, and from the companion cell, it diffuses into the sieve tube element. Okay. All right, just 3 marks. That can you see that? Okay. This mentioned, I want to pronounce it. Let's see. Silidine LL37 is a protein composed of 37 amino acids. Wow. The sequence of the 10 amino acids in the primary structure of catalycidine LL37. Okay, that's the sequence. We have DNA triplet in the non-transcribed strand in the gene that code for the first 10 amino acid in the primary structure of catalycidine LL37. So you can see that, guys. So this is the first 10 amino acid. Amino acid 1 is leucine, that's position 1, and the DNA triplet that codes it is CTG. You see that order. Okay, let's see. So it's table 5.2 shows the triplet of bases in DNA and the amino acids for which they code. Okay, now that's still this table. You can see second base, first base. All right, so you can see that it's simple. This is genetic dictionary. So if you say T, that is this. TTC, all right, that is this, and that code for phenyl, the amino acid. Okay, the same thing. So this is first base, second base, and that represents third base. All right, let's see what they want us to do with that. Right now, the question says mutations of the DNA-based sequences in a gene can affect the primary structure of proteins. Use the information in table 5.1 and table 5.2 to suggest the effect on the primary structure of catalycidine LL37 of the substitution of the base T with the base A. So T substituted with base A in the middle of triplet at position 5. So if we go back, what is that again? Okay, look at it. Okay, in the triplet at position 5. So A at the middle of that. So let's go back, but I want you to try and see what the impact it will have. So this is position 5, right? So this is the middle, right? If you remove this T and put A, you get what is going to happen? If we put A, that means we're going to have, instead of TTC, we'll have what? TAC, TAC. Alright, we're going to have TSC. That's mutation, base substitution. Okay. base substitution. So how will it affect the amino acid? Remember that TTC, which amino acid is TTC? Let's come here. TTC is phenyl, but after mutation it changes to TAC, which is this. TAC is is serine, right? Yeah, no, okay, is not serine, it's tyrosine. Okay, so that means that there's a change in amino acids from phenyl to serine to tyrosine. Okay, TTC is this, which is phenyl. TAC is this, which goes for this, right? That will be the impact of that, right? Okay. Look at that. That position 5. It says TTC to AC changes from phenyl to at position. So that is it, guys. 1 mark. Now the deletion of base T in the triplet at position 2. So if we delete T in the triplet at position 2, so what effect will it have? Let's go delete T, guys. This is position 2. If you delete this, okay, I want to pause the video and try it out. If you delete T, what do you think will happen? So that means that if you delete T, we're going to have C, G, right? But this G on the third will join them. So it's going to be C. You see, it's altering the amino acid sequence. That's frame shift. So we're going to have G, T. All right, but it needs to be completed. So this is going to join them, which is G here, right? C is altering the amino acid sequence. We're going to have A, because the first G is gone. Okay, move to the other base triplet. We have AT. Okay. Let me quite remove the other question we solved. So yeah. So you can see. Okay, let's just move because we've done with it. We'll deal with this. Okay. All right. So stay with me, guys. So we're gonna have A. This T will join. Okay. Yeah. Can you see T? So that way. So we have T and C. Okay. And then we're going to have this T will join the first T, there will join them. It's going to be TCC. So we're going to have TC. The next C in the 7th position will join them to be TCC, and then we're left with the other two, GG, and the other A will join. Do you get? So this is frame shift. It alters the base sequence from the point of mutation to the end. So you can see what is happening. This is what we call frame shift, and this result to what we call nonsense mutation. It's sure going to produce a different protein. So you can see this is going to be C, T, right, and then we're going to have A here. Do you get? So just have the last one will not be complete, AA, okay, because one base is deleted. So you see from this second base, second position, I mean to say from here there's oxidation. So from here, now remember the second one is leucine, but let's go change now what is CGG. Let's go change CGG. If you go check CGG is you can see that is arginine. So it has changed from leucine to arginine. So the second position here is no longer leucine, it's now arginine. Okay, let's check. The third one here used to be glycine. Let's check. So GGT to GTG. Let's check GTG. GTG. Yeah. So valine. So it's now valine. So it changed from glycine to valine. Can you see? Now fourth position is ASP. GAT is now ATT. All right. So asparagine to what? Let's check ATT. ATT is okay, leucine. Okay, it has changed from asparagine to ILE. So it alters the amino acid, okay, the primary structure of the protein from the point of mutation. So you see what happens. See what the answer says. So new sequence, right? See new sequence it lists for us here. All right, after first amino acid, of course, gives us after the first one. All right, the rest from position 2. So we're going to have, of course, also we're going to have shortened polypeptide. Remember that the last one, okay, the amino acid, the base triplet is not complete. All right. Okay, they say the insertion of base G between G and T in triplet at position 3. Insertion of base G between G and T. So we'll go to position 3 and insert base G between G and T. So I want you to try that, okay? Yes. Let's go back to that. Yes, we should go back. Yes, let's look at it. Yeah, insertion. Can you see? Let's go and insert. Yes, this is it. Insert base G between G and T, right? But first of all, let's clean, wipe off what we've done and see what happens from here down. Okay, yeah, let's clean it up. Okay, yeah, when you insert it, what do you have? What do we have left, guys? What do we have? So if you insert it, if you insert G here between these, so we're gonna have, all right, triple G, 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 G. Okay, that T will go to the next one. It's gonna be T, right, G, A. You get? So this T will move to the next one. Okay, so it's gonna be T, T, T. Okay, just like that. So the next one is going to be C, T, T. But let's see. All right, you see that it's altering base sequence again. That's frame shift. This is frame shift, guys. But let's observe. Let's check what has happened so far. Come on, guys. Let's check it. Okay, now from this position 3, because they said what happens if you insert G in between G and T. So we inserted in between G and T, and then we have GGG. So this T moved over to here. Forms this is this T is this T. So we're gonna be T G A. So from here, GGT is glycine. Let's check GGG. GGG triple G. Okay, look at okay. GGG is still the same glycine, right? Yeah, the same glycine. So that means GGT and GGG, the same amino acid, all right? Okay, let's go to position 4. We have TGA, from GAT to TGA. Let's check TGA, TG, okay? A, that's stop codon. Stop codon. So that means that this shows stop codon. When it shows stop codon, then translation stops there. So we now have shortened polypeptide. Polypeptide will have only 3 amino acids: lysine, lysine, and glycine. Here is stop codon. So no more amino acid will be added. So you see the impact of that mutation. Okay, the impact of that mutation. Look at it here. The impact. Yeah, I think that's it. Yes. So you see, peptide 3 amino acid in length, shortened polypeptide, stop codon in position 4. Third amino acid is still glycine. Yes. So all right, just 1 mark for all of this work. The genetic code is described as universal. Explain why genetic code is described as universal. Okay, I want you to try it. Why is it described as universal? It's because, pause the video and try it. It's the same. Genetic code is the same in all organisms. So if a particular base triplet codes for an amino acid, is the same for all organisms. Let's not waste time. Okay, it's the same, similar in organism. And each triplet code for some amino acid in all organism. 1 mark. Use table 5.2 to explain why some mutations have no effect on primary structure of a protein. All right. So if you go back to that 5, all right, pause the video and try it. 5.2. Why some mutations have no effect on primary structure. I think we've seen it before. Okay. If you go back, you can see. Look at that table. Where is that table? Yeah, come with me. Yeah, table 5.2. All right. You can see that an amino acid can be coded by more than one base triplet. Okay, look at phenyl, more than one base triplet. Okay, look at leucine, coded the same amino acid coded by, of course, 4 base triplets. So if there was a mutation and then we change this to this, okay, what happens is that the same amino acid is coded. Do you get? Right? So you can see. So if mutation, especially the third, you can see that the third position bases, when you change them, they'll still give you some amino acid. Okay. All right. So that is it, because that's called degeneracy. One amino acid can be coded by more than one base triplet. Okay, that's it here. Let's see it. So we've done that. Okay, that's the question here. Explain using why some mutations have no effect. Why? This is it, guys. Right? See it? Most amino acids have more than one triplet. Okay. Yeah, you can check. Genetic code is degenerate. That's okay. Yeah, I can mention them. That is why out of 64 base triplet, they can only code for 20 amino acid. Alright, because some of them, more than one codes for one amino acid. And those some base triplet do not code for any, alright, amino acid. Okay, like stop codon. Alright, look at it. The nuclei of plants produce small lengths of RNA known as microRNAs. MicroRNAs in gut cells have been shown to prevent the synthesis of some proteins. The gut cells of drought-tolerant tomatoes plant produce more microRNA molecules than the gut cells of the non-tolerant plant. MicroRNA molecules do not prevent transcription but interact with messenger RNA. Suggest how this microRNA can interact with messenger RNA. Okay, microRNA interacting with messenger RNA, mRNA, to prevent the production of proteins in gut cells of S-lycopassion. So I want you to pause the video and try it. Alright, pause the video and try it. Alright, remember they said here that microRNA molecule do not prevent transcription, but it interacts with messenger RNA. So that means it can bind to messenger RNA. The bases of microRNA are complementary to the bases on messenger RNA. So immediately after messenger RNA is formed by transcription, microRNA binds to it by complementary base pairing. Okay, when it binds to it, there are consequences to that, and that's what we're looking at here. So just how this prevent the production of protein. Now one of that is it prevent production of protein. How? So when it binds to the messenger RNA, messenger RNA will become too big to leave the nucleus because it's single-stranded, right? But now, binding to another single-stranded RNA will make it double-stranded. But the big thing there is that once it binds to messenger RNA will not be able to bind to the small subunit of ribosomes. Okay, so apart from that, transfer RNA can no longer bind to it, and then primary structure of course proteins cannot be formed, because if transfer RNA carrying amino acid cannot bind to a messenger RNA, then peptide bond cannot be formed between transfer RNA. Okay, and then transfer RNAs cannot be reused. Is that okay? Look at it. So this is going to bind to this because two of them, all right, of course they interact, so forms hydrogen bond between their bases. Bases in micro are complementary bases on this, all right, and messenger RNA cannot bind, all right, to small subunit of ribosome. Okay, accept prevent ribosome from moving along, so ribosome cannot move along mRNA. Okay, so it stops mRNA from undergoing, acting as template for translation. So anti-codon of transfer RNA cannot bind, cannot bind to codon on mRNA because microRNA is already binding to mRNA. So that's a big problem. Not at all amino acid are brought to ribosome. Of course, no amino acid brought a ribosome. Of course, that's what happens, and of course, primary structures are not formed. This is 8 marks, guys. Is it 8? No, it's not 8. Alright, so blood cells are formed from tissue stem cells in the bone marrow. These bone marrow stem cells go through a number of mitotic cell cycles to form fully functioning cells. Fig 5.1 shows the 3 stages of the cell cycle. See the interphase mitosis cytokinesis. Says the activity of genes changes during the mitotic cell cycle. When genes are being expressed, the cell produces many messenger RNA molecules and ATP molecules. Okay. So explain what is meant by gene. Okay, pause the video. What is a gene? What is a gene? Pause it. A gene is a section, a length of DNA that codes for one protein, one polypeptide. Okay, sequence of DNA nucleotides forms part of DNA nucleotide, length of DNA coding for a polypeptide or a protein. 2 marks. All right, you can see that. Say the main stages of cell cycle in Fig 5 during which say name it, during which most mRNA and ATP is formed. Okay, so what are the stages in cell cycle? Name the main stages, all right, of cell cycle in Fig 5.1, which most mRNA and ATP is formed. Look at it now. So where is mRNA formed? That is where protein synthesis takes place. Of course, it takes place in interphase. Okay, so they're asking us to mention the stages of interphase, but you can stop and attempt this question. Okay, so interphase has how many stages? Do you remember? It has if you remember the cell cycle, right? You see that interphase, if you draw it this way, you will see, if you remember, this is mitosis, that's M. Okay, that's M, like mango, mitosis, right? This is cytokinesis. Now we have the first stage here of interphase, which is G1. Okay, this is S phase, this is G2. So interphase has 3 substages. So and then of course mRNA is made there in these stages, because this is growth 1. So in growth 1 have, okay, ATP is made here, the cell grows bigger, of course protein synthesis occurs, which include transcription and translation. So transcription is synthesis of messenger RNA is made here. So in G1 we have ATP, of course, new organelles formed, the cell grows bigger, okay, more proteins made that will be needed here. So transcription, translation occurs. This is semi-conjugated replication of DNA occurs in S phase. G2, protein synthesis occurs here, mRNA made. Okay, and of course we have a lot of checkpoints here going on. Okay, DNA is proofread here to determine whether there are possible genetic errors in G2. Organelles like centrioles replicate. All right, and all of that. Okay, so let's see what we have there. See, that's interphase. That's interphase. All of this happening. Fig 5.1 is an incomplete diagram of ATP. So you can see that. So the question says, complete the diagram of the ATP molecule. Write the name of the base in the space provided. Write the name of the sugars in the space provided. So what are the names here? This is ATP. Pause the video and try it. This is a purine. That's adenine. Okay, is a purine, so it's double single. Okay, double ring structure. Double ring. This is pentose sugar, but it's called ribose. That's phosphate. Okay, so you can see 2 phosphates, of course, added to existing phosphates. So once you add it here, it completes it. But B, this is base adenine. Sugar is ribose sugar. Okay, 3 marks. It's an incomplete diagram of ATP molecule. Okay, let's see the same question. The base has a double ring structure. State the term for a base that has double ring structure. That is a purine. Purine. 1 mark. Fig 1.2 is a diagram showing a stage in protein synthesis. Yep, this is interesting. Okay, so this is protein synthesis. You can see this is, I want you to try this question, guys. Name the stage of protein synthesis that is shown in Fig 1.1. Which stage is here? All right, which stage? Of course, is translation. All right, that is synthesis of polypeptide. Okay, so Fig 1.1. Okay, the same. Okay, let's look at the second question. Identify A, B, and C. So what is A? this particular molecule here. Okay, what is it? What is B? Okay, what is C? Pause the video and attempt it. A is messenger RNA, B is transfer RNA, C is polypeptide chain. Okay, 3 marks. Fig 1.1 is a diagram showing a stage in protein. I think that the same here. So next question, state the base sequences at D and E. So yes, pause the video. What are the base sequences at D and E? You can see D. Look at it. All right. This is the anticodon of transfer RNA. This transfer RNA, okay, is complementary to this is codon on messenger RNA. Okay, this is messenger RNA. So this is going to be complementary to A. So D should be AAA. Now if you look at E, okay, this is C. Here will be G. Okay, this is A. This is going to be U. This is C. This is going to be G. So it's going to be GUG. Right? Okay. Mutagenesis is a process that leads to a change in the amino acid sequence of proteins. Scientists carry out mutagenesis to investigate the importance of particular amino acid in protein structure and function. All right. Outline how changing one amino acid in beta globin or polypeptide of hemoglobin may change the structure and function of molecule of hemoglobin. So I want you to pause this video and try it. How does changing one amino acid, okay, in beta globin polypeptide? Remember, the amino acid that is changed here is the amino acid we call glutamic acid. So glutamic acid is replaced with valine. So glutamic as changing amino acid. Anytime you are dealing with mutation, just remember that change in amino acid, change in base triplet will result to change in amino acid, which causes a change in R group and causes a change in the interaction between R groups, and that causes a change in the secondary and tertiary structure. And then once tertiary structure is changed, it causes a change, all right, that affects the function of the protein. So the protein here is hemoglobin. So valine replaces glutamic acid. So we now have hydrophobic R group. Okay, that changes the structure of hemoglobin, changes the tertiary structure of hemoglobin, and then this means that hemoglobin, it will affect the function of hemoglobin. It transports less oxygen, has lower affinity for oxygen, so it becomes less saturated with oxygen. Look at it here. Different amino acids will have different R group. Yes. So valine has hydrophobic, while glutamic. So you see that glutamic is okay replaced to valine. That's typical of what's happening here. Okay, may prevent formation of bonds. So some bonds don't form here between R groups, and because of that, new bonds are formed. Because hydrophilic R groups cannot form hydrophobic interaction. So new bonds are formed between the R group. There's a change in secondary tertiary structure and even quaternary structure. This quaternary structure, change in global shape of hemoglobin, a different fold of beta globin. You can see that. So they say reject quaternary is referring to. Now quaternary refers to right. It refers to more than one polypeptide chain. We are dealing with one polypeptide chain here. Okay. All right. So any further detail, that's a change in primary structure. Interaction between may change. You can see because there's a change from polar R group to okay. So let's see what happens to the function. It changes the ability to bind hemoglobin. Okay, it reduces the ability because the tertiary structures change. It has lower affinity. Okay, for change in percentage saturation of hemoglobin, change in affinity for oxygen and carbon dioxide. So more or less oxygen constant. Transported 4 marks. Comes out every time. 3.1 is the diagram of monomer of nucleic acid messenger RNA. You can see that. Okay, said name DEF. So name it for us quickly. D is a pyrimidine. E ribose. That is a phosphate group. Okay. Okay. So telomeres are repeating sequences of base located at ends of DNA molecules. These repeating sequences do not code for protein. The enzyme telomerase ensures that telomeres do not shorten each time DNA is replicated. Fig 3.22a shows the end of a DNA molecule during replication. DNA polymerase cannot attach to the region labeled X, so it cannot complete the synthesis of new strands without the action of telomerase. Okay, now telomerase, so it cannot complete it without the action, so it needs telomerase. Telomerase synthesizes additional lengths of DNA that are added to the telomere. These additional lengths are used by DNA polymerase to complete the process of replication. Okay, so you can see that. So see, this is the end. So they say DNA polymerase cannot attach to the region labeled X. It cannot, so it cannot complete it, right? But telomerase, it says, cannot complete synthesis of new strand without the action of telomerase. So telomerase synthesizes additional length of DNA. Okay, it's gonna synthesize additional length of DNA here, right? Okay, so let's see how it does that. Okay, it says an enlarged view of region X to show the action of enzyme telomerase. So look at that. See the action of enzyme telomerase. This is telomerase enzyme. Can you see? So it uses RNA template to make DNA. This is DNA nucleotides. There's no uracil here. So using it to make DNA. Okay, let's see what they say. Okay, telomerase contains a short length of RNA that acts as template for the synthesis of DNA as shown in Fig 3.2b. Okay, explain how a molecule of telomerase synthesizes additional length of DNA. So how does this? Pause the video. So you have to come here, pause the video, and then try to explain this. Okay, you can see that. See this. Telomerase enzyme binds to the DNA. All right, using its active site. This is active site, right? Okay, but it also uses the template, RNA template. Okay, so RNA template to align to make DNA, okay, strands. So then RNA is used as a template, all right, to make DNA. So this RNA, the telomerase enzymes, allow this activated free nucleotide to bind to, first of all, they bind to their complementary base pairing, okay, and then phosphodiester bond is formed between them to form a strand. So see what I'm trying to say, guys. If you remember how this works, if you remember, okay, for example, there's this one here. So the one that will attach here is G, okay, the one that is carrying, okay, this is C, attaches with, right, G again. This A will have T, okay, so this is T. Yeah, that's nucleotide, right, has T. This has, okay, this is gonna be T, right, so that uracil will have A there, okay. So this is what the telomerase enzyme does. It aligns DNA nucleotides, right, to bind to the complementary bases, right, on the RNA template strand, and then it now forms phosphodiester bond between them. Okay, this is phosphodiester bond that forms between them to link them up to form a strand of DNA. Can you see? All right, so in other words, it's acting like a reverse transcriptase, which uses RNA as a template to form. So this is how it aligns them and let them form hydrogen bond with the complementary bases, and then form phosphodiester bond between them. Do you get? Let's see what the masking says. See, template strand binds to, yeah, the region X. That's where it binds to, in active site of telomerase enzyme. DNA free nucleotide pair with or align with RNA complementary base pairs, like what we said. Phosphodiester bond forms between DNA nucleotides. You can see, treat as neutral. Okay, yeah. Telomerase moves in the direction, yeah, of the arrow. Hydrogen bond between bases. Telomerase act as reverse transcriptase because that's what it does. So thank you guys for hanging on. This is Shedro. Okay, so see you in our next video. I wish you best in your coming exam. All right, watch all our topicals. Bye for now.